Makita ra class?
Yes, sir.
Okay. So it's 9 already, then I'll start the ratio. Okay. So this is just a short rationalization of the previous long quiz. So we'll start though. So for items 1 to 10, you have there a case. Wales, a 75-year-old man lived in a retirement village. He received periodic medical checkup and was relatively good. During winter, an outbreak of influenza affected many residents of the complex, even though all received vaccinations. The man developed a progressive lower respiratory tract illness with symptoms including abrupt chills, high fever, productive cough, and chest pains. Sputum cultures revealed large number of gram-positive diplococci, which are also found in the blood samples. Therefore, class, the patient is having pneumonia plus bacteremia, possible for pneumonia and possible for bacteremia. Chest X-ray showed lobar consolidations. During his hospitalization, he was given with penicillin G antibiotics, and later the man's condition deteriorated, ending with his death. So the death is usually accompanied by the immune responses, no? So question 1, the organism observed in patient's sputum sample and blood probably belongs to the following genera. So you have there a clue, no? It's gram-positive diplococci. The gram-positive diplococci, so you already have idea, Staphylococcus is gram-positive, but it's not diplococci, it is cocci in clusters. Neisseria, although it's diplococci, but it's not gram-positive, it is gram-negative. Enterococcus is gram-positive. Some may be gram-positive diplococci, but mostly class, they are arranged in gram-positive in chain. Okay, so the answer there is your Streptococcus. The organism strict on the blood agar plate would produce. Okay, since you have there an idea, number 1 will give you an idea, class, that the patient is having pneumonia or respiratory disease. So think of which Streptococcus is possible for respiratory disease. So ano man yung mga Streptococcus species na may possible for pneumonia? So you may think of Streptococcus pneumoniae and Streptococcus viridans. So the organism strict on a blood agar plate would produce alpha hemolytic, beta hemolytic, no hemolysis, or do not grow. Okay, so since you will be thinking of 2 possible organisms, your Streptococcus pneumoniae and Streptococcus viridans are both alpha hemolytic class. Okay, yung Enterococcus may be alpha hemolytic, beta hemolytic, as well as no hemolysis at all. So, third question, vaccines against the organisms are prepared against the, remember class, no, vaccines are usually prepared only sa ilahang cell wall. If the organism is, cell wall, if the organism is capsulated, for example, Streptococcus pneumoniae, Neisseria meningitidis, Haemophilus, mga capsulated na sila class. So therefore, pag capsulated organism, think na maugid na ang source sa ilahang vaccines. Okay? So for pneumococcal or pneumococcus class, capsule na siya ilang source sa ilahang vaccine. Diba? For the pneumococcus, you have the PCV 13 and the PSCV na 23. PPSV 23 rather. Number 4, the toxin isolated from animals infected experimented with the organism is Since you have their Streptococcus class na answer, ang only toxin na ma-release ni strep diri sa choices is the pneumolysine. Okay? Shiga toxin is released by Shigella. Diphtheria toxin is released by Corynebacterium diphtheriae. The LOS toxin, that's lipo-oligosaccharide toxin, is released by most of the gram-negative organism. So pneumolysine is the best possible answer for this. Okay? Your pneumolysine may act as antiphagocytic. It also reacts with your cytokines. And also your pneumolysine is also responsible why your blood agar plate will turn into alpha hemolytic. Okay? So, ito yung responsible bakit naging alpha hemolysis yung blood agar plate. Number 5, which of the following is not, no, it's not correct. Take note, not correct statement regarding blood culture. So for this question, try to remember your previous discussion about blood collection. So a collection of 10 to 20 ml per culture for adults, that's correct. 2 to 3 bottles for blood culture is recommended as optimum. 2 to 3 so that we can get more organisms, no? In case we miss that in a one culture battle, so at least may backup pa tayo. Also, we need to perform aerobic and anaerobic culture. So for adults, the one bottle is for aerobic and the other one is anaerobic. That's correct. Letter C, volume of blood cultured is more critical than timing of the blood culture. So that is correct also. We need to have more volume of blood rather than the timing of the culture. Okay, hindi kasi necessary class na at the peak, no, of the fever ang mag-collect ng blood culture. The blood culture should be collected either before sang antibiotics ideally or after sang antibiotics, pwede pa din kasi yung blood culture bottles nyo, remember, meron siyang antibiotic removal device. So pwede siya na mag-collect kahit meron pang antibiotic. So, importante talaga i-follow yung blood because if lesser value ang blood na mas marami pa yung anticoagulant, meaning wala po na siya ma-collect na organism class. Letter D, blood drawn for culture may be allowed to clot. So, hindi pwede mag-clot yung nakuha mo na blood. Remember, class, your blood culture bottle or your yellow bottle, yellow yung color ng blood culture bottle. Usually, for sa lahat, no, if CLSI guidelines ang ma-follow. But on practice, class, lain-lain na silang color. You will not see or you will not accept when you become medical technologists, you will not accept class if ever makita mo na may blood clot ang blood culture bottle. Wala dyan na dapat blood clot because again, your blood culture bottle contains still anticoagulant and that anticoagulant is your sodium sulfonate. Okay? So review. What is the initial biochemical test that should be done by a medical technologist to identify the suspected species? Since you already know alpha hemolytic siya class, the very best answer for this is your Taxo-P. Okay? Bile isquilin is commonly used for beta hemolytic organism. Coagulase, if you want to differentiate strep from staph. Oxidase is usually done if ever it's gram-negative organism. So Taxo-P, remember, is your optokin test. Okay? Or your ethyl hydrocarbon hydrochloride na antibiotic. So possible result for Taxo-P is more than 14 mm. Number 7, what is the mechanism of antibiotic mentioned? No, the antibiotic mentioned here is penicillin G. So penicillin class is under beta-lactam antibiotics. So penicillin, is not under beta-lactams. It's separate, but another group of cell wall inhibitors antibiotics. So ito yung answer. Basta lahat ng mga penicillin at saka beta-lactam drugs class are all cell wall inhibitors. So based on Bartlett's criteria, what is the ideal sputum sample for culture? So here, more than 10 epithelial cells per field, more than 25 pus cells per field, less than 10 pus cells per field, and less than 25 epithelial cells per field. So ideally, or the best answer class should be, it should have more than 25 or polynuclear cells or your pus cells. Kasi meaning there is inflammatory response. If more than naman yung epithelial cells class sa sputum sample, meaning that is not a true sputum sample, it's only saliva. So pag maraming epithelial cells compared to your pus cells, saliva lang yan. Pero pag marami yung pus cells or your neutrophils, than your epithelial cells, then possible that is sputum sample. So if ever you'll receive sample na nakasaliva rhizia class, reject that sample or tell the patient to collect another sample, tell them how to collect sputum sample properly. But when sa internship nyo class, hindi kayo pwede mag-reject directly. You just ask first your medtech in charge kung saan kayo na-assign. Number 9, all are possible complication of the etiologic agent except, okay, since you already know that this is Streptococcus pneumoniae, your Streptococcus pneumoniae class can cause at least 4 major diseases. So the mnemonics is MOPS, M-O-P-S. Again, the mnemonics is MOPS. So M-O-P-S. No, this is the mnemonics for the disease of Streptococcus pneumoniae. M stands for meningitis. O stands for otitis media. P stands for pneumonia. And S stands for sinusitis. So among them, the endocarditis mo'y wala. Okay? Endocarditis is usually caused by Enterococcus. It also caused by Streptococcus viridans and also your Staphylococcus aureus. Okay? So yun yung mga possible causative agent ng endocarditis. Number 10, what is considered as confirmatory test to identify the suspected agent? So you have their optokin test, bacitracin test, Killang test, and coagulase test. So optokin test and bacitracin test class are just initial testing. Okay? But in some laboratory, pag mag-optokin sensitive na siya, mustop na sila. But there are still testing, confirmatory tests that will really give you a presumptive diagnosis na it's really Streptococcus pneumoniae. And one test is your Killang's test. Okay, or your Neufield's Killang test. Neufield's Killang test class is a capsule na testing. So for this, positive result is the swelling of the capsule. Since most of the Streptococcus species are not capsulated, so only your Streptococcus pneumoniae may appear positive for Neufield's Killang's test. Question so far for 1 to 10? Wala. Okay, let's proceed to 11 to 16. 11 to 16 is another case. It's a male patient with culture positive gonococcal urethritis. I hope you're writing class how while I'm discussing because some of this may comes out during your P2 exam. So, a male patient with culture-positive gonococcal urethritis was treated with 5 million units of aqueous procaine penicillin G intramuscularly, plus probenecid 1 grams orally. 8 days later, he returned complaining of similar symptoms consisting painful urination but less urethral discharge. So question 11, what is the suspected primary causative agent of the above infection? So on the infection, you have their culture-positive gonococcal urethritis. That's the clue already. So among these 4 choices, only Neisseria gonorrhoeae class can cause gonococcal urethritis. Chlamydia trachomatis can cause non-gonococcal urethritis as well as Candida albicans, but E. coli will not cause any non-gonococcal urethritis. So, si Neisseria gonorrhoeae gyud ang only best answer for this. Number 12, if the causative agent responsible for the above infection were gram-negative Diplococcus, what biochemical test is used to confirm the above gram stain finding? So, the choices, coagulase, catalase, oxidase, and indole testing. So, for this, the best answer is oxidase test. So most of the Neisseria species are oxidase positive. Okay? Although some of them are catalase positive, but catalase positive also ang other bacteria. So ang pinaka-best answer diri is oxidase test. What is the reagent of the biochemical test used in number 12? Okay? So the reagent for oxidase test class, take note, that is 1% tetramethyl paraphenylene diamine dihydrochloride. So take note of this, ha? Because this will usually come out class sa mga board exam recalls. 3% hydrogen peroxide is the reagent for your catalase test. COVAX is for your indole testing. Serum is not usually used as reagent, but it's plasma for coagulase test. Diba? Na-perform na ninyo. Number 14, what is the possible virulence factor of the primary pathogen for the above infection? Somatic antigen, somatic pili, capsular antigen, and flagellar antigen. So the primary virulence factor for Neisseria class is pili, no? And that pili is somatic or common pili. Your Neisseria gonorrhoeae do not have capsule, they do not have flagella, and somatic antigen is not really an answer. Wala talagang somatic antigen class, somatic pili. Again, another question, what's the mechanism of action of penicillin? So you already know sa number 10 kanina that penicillin inhibits cell wall. So that's still the same answer. Number 16, in doing carbohydrate utilization testing, what sugar is fermented by the primary etiologic agent? Remember class, no, pag gonorrhea, di ba gonorrhea, pag gonorrhea class, G there, always think of glucose. Okay? Sila tanan class, most of the Neisseria species were ferment glucose, but only Neisseria gonorrhoeae, glucose ra na yung i-ferment. The rest na sugar, negative na siya. For glucose and sucrose class, usually si Zika na siya, si Neisseria Zika. So si Zika is their sucrose. Your glucose and lactose, you have another species of Neisseria, which is your lactamica. So, glucose and lactose, that is for Neisseria lactamica. Neisseria sicca and Neisseria lactamica class are opportunistic pathogen. These are commensal organism. So they are part of the normal flora lang. But they can also cause diseases if na a chance. But these two are just normal flora of the upper respiratory tract. So if ever glucose and maltose class, so you think of meningitis. Again, gonorrhea, think of glucose only. Zika, glucose and sucrose. If glucose and lactose, that's lactamica. And if ever glucose and maltose, that is meningitis. Number 17, which of the following is a selective medium for Neisseria gonorrhoeae? Okay. So, MHIA or your Muller-Hinton infusion agar is not a selective medium. This is usually used as a special medium for antibiotic testing. Your CAP is not a selective medium. It's more of enrichment medium. And, diba? And MTM stands for Modified Thayer-Martin agar. Modified Thayer-Martin because this is a selective culture medium for Neisseria species because it contains different antibiotics that inhibits the organism. So it contains vancomycin that will inhibit gram-positive organism. It contains colistin that inhibits other gram-negative organisms, particularly your gram-neg bacilli. It also contains nystatin antibiotics, which inhibits fungal infection. And this become modified class. This is different from your ordinary Thayer-Martin because another antibiotic is added, which is your trimethoprim, that will inhibit proteus growth. Because remember, diba, in your case, proteus is a swarming colonies. Okay? So if ever mag-swarm siya class, matabunan to ang mga possible organism na motubo kay tungod kayang swarming nga appearance. So para dili ta maka-isolate og swarming colonies, we need to add another antibiotics in the form of trimethoprim na antibiotics. Okay? Modified Thayer-Martin is specially used class if ever we will collect fecal swab. Okay? So mga swab from the lobot. So kanatong gamiton kasagaran. Blood agar plate is never a selective medium, remember that. Because it will allow growth of almost all organisms. Number 18. Staphylococcus epidermidis is recovered from which of the following sources? Remember, class, your Staphylococcus epidermidis is the causative agent of most medical devices. So it can cause bacteremia that is associated with medical devices. If ever the patient has prosthetic heart valve, intravenous catheter, and urinary tract infection. So the answer is all of the above. Because your Staphylococcus epidermidis class will easily form biofilm. So kaya ma-adhere siya or ma-attach siya sa mga medical devices. Number 19, bacitracin A disc are used for presumptive identification of which group beta hemolytic. So you already know this. Bacitracin is a disc that will identify group A strep or your Streptococcus pyogenes. Which Streptococcus grows on 6.5 salt broth, bile esquilin positive, and ferments sucrose and trehalose, but not on arabinose? The correct answer is Enterococcus faecium. Your Streptococcus mutans and Streptococcus pneumoniae are not bile esquilin positive. Your Enterococcus, so for this one class, 6.5 bile esquilin positive ang imong i-think ani is Enterococcus. Since you have two Enterococcus nga choices, you are further given no sugar identification. So if ever the sugar fermented are only sucrose and trihalose, then that is phasium. But if ever class fermented ang tanan, sucrose, trihalose, and arabinose class, then that is fecalis. Again, pag 6.5, so broth, bile is killing positive, think of Enterococcus. Since you are given another sugars, ferment sugars, sucrose and trihalose only, then that is phasium. Pag ang tulo ang na-ferment, then that is Enterococcus fecalis. 21. Which test is used to differentiate Viridans streptococci from group D streptococci and Enterococci? So you have there, as I mentioned, bile is killing test. Bacitracin test, this test, CAMP test class, and hippurate hydrolysis test are used no for the determination of beta hemolytic strep. Oy, paki-erase sa kanina nagsulat, sa kanina nagsulat. Number 22, which of the following enzymes contributes to the virulence of Staphylococcus aureus? So the choices, urease and lecitinase. Urease and lecitinase is not released, no, nor part of the Streptococcus aureus na virulence factor. Hyaluronidase and beta-lactamase can be. Lecitinase, again, is not part. Catalase, pwede. Cytochrome oxidase. Cytochrome oxidase is not part of Staphylococcus aureus. So, ito yung best answer. Hyaluronidase class, di ba, will, the organism has the ability to lyse the hyaluronic acid that is found on the connective tissue. Yung hyaluronic acid, ito yung parang glue ng mga connective tissue. So, si Staphylococcus aureus, kaya niyang gibain yung parang tight junctions ng connective tissue para makapenetrate siya downward. Yung beta-lactamase naman, ito yung mga naproproduce na mga Staph aureus pag methicillin-resistant staph. So, they are capable of making any beta-lactam drugs that is that they resist no any beta-lactam drugs. So hindi pwede yung penicillin as treatment of choice if ever MRSA yung Staph aureus. Slime production is associated with what Staphylococcus species? So the answer is Staphylococcus epidermidis. So as I said kanina, si Staphylococcus epidermidis will easily form biofilm kasi dahil sa kanyang slime. Bacterial species that can be described as oxidase positive, glucose positive, maltose negative, sucrose negative, and a major cause of venereal disease. So kanina na-mention ko na to. Ang positive lang is glucose positive. So you have there Neisseria gonorrhoeae. Okay? Hindi naman pwede class ma-answer si Streptococcus pyogenes as Streptococcus viridans kasi the question is cause of venereal disease. Yung dalawang ito, si Strep pyo at saka si Strep viridans, will really not cause venereal disease. So ano yung mga venereal disease? Ito yung mga syphilis, no? Chlamydia, yun yung mga venereal disease. Mga sexually transmitted infection mostly. 25. Throat culture was taken from a 6-year-old boy with a gray pseudomembrane covering his oropharynx. A catalase-positive organism was isolated on cystine tellerite and subcultured on Tensdale medium, where it grows black colonies with brown halos. A gram stain was performed on these colonies. Which of the following cellular morphologies was most likely seen? Okay. The clue here, class, is 1, grayish pseudomembrane oropharynx. If you read this one, na grayish pseudomembrane oropharynx, think of one organism. This is only associated with Corynebacterium diphtheriae because your grayish pseudomembrane, class, is a symptoms, no, or a sign rather of diphtheria. This is the hallmark of the disease of diphtheria. Another clue is the growth on cystine tellerite and Tensdale medium. The cystine tellerite and Tensdale medium, these are common culture medium, class, or culture media used to facilitate growth of Corynebacterium diphtheriae. Okay? Usually, pag Corynebacterium diphtheriae, ang growth is black colonies. So, since you have their idea nga this is Corynebacterium diphtheriae, so you look for gram-positive bacilli because your Corynebacterium diphtheriae is a gram-positive bacilli. So, eliminate those that are not gram-positive bacilli sa choices. So, you eliminate letter B and you eliminate letter D. So, you have letter A and letter C na lang na mabilin. Your Corynebacterium diphtheriae, di ba? In your readings, these are arranged in picket fence and these are pleomorphic. So, your letter A, branching bacilli, never yun na siyang correct answer because when you say branch, connected siya, daghan. But when letter C, that's irregular club shape, pwede siya. Kasi, meaning pleomorphic. Dili siya connected, dili hiwalay-hiwalay imuhang pagkabasilay sa iyaha. So the best answer for this is letter C. Okay? Example, by the way, of common gram-positive branching bacilli are your nocardia, actinomycetes, yun yung mga branching bacilli. And if you grow this on culture medium, class, makita mo siya nga mura pud siya ga branch iyang mga colonies. Okay, so remember again, Corynebacterium diphtheriae, hallmark of the disease, formation of pseudomembrane. And iyang gram staining is gram-positive bacilli with irregular club shape, or these are pleomorphic bacilli, or these are the bacilli na ginatawag na to yung characteristics kay picket fence or Chinese letter in characters. Number 26. Which of the following sets provide best differentiation of Erysipelotrix and from Listeria monocytogenes? Okay. Take note or remember, class, that among the gram-positive organism, particularly bacilli, only Erysipelotrix class produce hydrogen sulfide. Okay, so meaning siya ra ang positive for hydrogen sulfide or with H2S. The rest na mga gram-positive bacilli, wala. So ito talaga yung dapat mong tingnan. Okay? So hydrogen sulfide production, if nagani ni hydrogen sulfide positive or hydrogen sulfide production, then that would be Erysipelotrix. Okay? CAM test negative si Erysipelotrix, CAM test positive naman si monocytogenes. Isculin negative si Erysipelotrix, isculin positive naman si Listeria. So take note ha, kani ang key for Erysipelotrix na organism. 27. Characteristics of being lactose negative, citrate negative, urease negative, lysine negative, and non-motile. Okay? Remember, ang bisag makalimot ka ani class sa mga result, the clue here is non-motile. Okay? Which of the following here is non-motile? Remember the general rule of family Enterobacteriaceae. So, the general rule, di ba, sa Enterobacteriaceae, all of them or all enterics are motile except two genus, your Klebsiella and your Shigella species. Okay? So here, si Pruchus, very motile because, again, it forms worm colonies. Salmonella is motile. Yersinia also is motile. So, here, Shigella dysenteriae, di ba part man si Shigella sa kung na-mention kanina, then that is non-motile. So again, duhara ang dili motile ha sa family nga Enterex. Si Klebsiella o si Shigella. So bahalag makalimot ka sa mga result aning biochemical test uban, the clue there is non-motile. Number 28, 28, pulmonary form of anthrax is known as your Worcester disease. Okay, take note of this. That's the pulmonary form of anthrax. Valley fever, this is caused by fungal infection, which is known as your coccidioides. Walking pneumonia, this is another infection which is caused by Mycoplasma pneumoniae. And farmer's lungs, this is often caused by molds na mga organism. 29, colonies of Listeria monocytogenes on blood agar plate most likely resembles colonies of, the correct answer here is Streptococcus agalactiae. Okay? Your Listeria monocytogenes class has the same characteristics, has the same biochemical test result with CAMP test. Ang kalahian lang is the formation of the result. Kang Streptococcus agalactiae, bowtie, iyang appearance kani, reverse, iyang CAMP formation. Number 30, which of the following reaction is a typical for E. coli? So, A, beta hemolytic on sheep's blood agar, B, colorless colonies, C, colorless on XLD, D, green colonies with black center on hecton enteric agar. So, B and C is never a correct answer, class colorless, because always remember class E. coli is a lactose fermenter organism. So again, pag lactose fermenter ang organism, no, it will appear sa Maconkey as colored. So dili gyud na siya ang correct answer. Letter D naman is rule out because of the word black center. Never mag-produce og hydrogen sulfide si E. coli. Okay? E. coli on an hectoid enteric agar may appear as colored colonies but never with black center. Black center is usually associated with Salmonella species. So, ang mabilin na lang is ang letter A, which is beta hemolytics colonies on a ship's blood agar plate. Number 31, colonies that are said to be resemble droplets mercury characteristics. So take note of this, no? Pag droplets of mercury that is associated with pertussis, Bordetella pertussis. I think this one class is bonus, no? Bonus siya because this is another, this topic will be discussed karon pang a week. So at least may one bonus. Number 32, Serenity test or Serenity test rather is used to diagnose which type of organism. Serenity test class is used to differentiate types of E. coli. Okay, so it gives positive results on enteroinvasive E. coli nga type. EHEC, that's hemorrhagic E. coli. ETEC, that's enterotoxigenic E. coli. EPEC, that's enterogenic E. coli. So si EHEC, di ba, ito yung parang bloody diarrhea. na E. coli. It will cause blood diarrhea. Same with Shigella. Si E-tech naman, ito yung mga associated with traveler's disease. Si E-pec naman, ito yung mga diarrhea associated with pediatric patients. So P there, remember P, pediatric patients. Number 33, all of the following enzymes are produced by Serratia except hyaluronidase is the correct answer. So the three are produced by Serratia species. Number 34, which Citrobacter species is only hydrogen sulfide, meaning only hydrogen sulfide producer. So among the four class, si Citrobacter frondiae lang ang hydrogen sulfide producer. Remember, pag Citrobacter species class, these are citrate positive. That's why ginatawag siya Citrobacter from the word citrate. Okay? Number 35, which of the produced species is the only source of OX2 and OX19 that agglutinates when the patient has rickettsial infection? Okay, so the correct answer is Proteus vulgaris. OX2 and OX19, you will learn more of this when we have the rickettsial discussion. The vector per bubonic plague. Bubonic plague is caused by Yersinia pestis. So the vector for that is rat flea. Okay. 37, dog ticks is different. Another organism, human louse. Usually dog ticks and human louse are associated with mga rickettsial species. Tsetse fly is more of protozoan species. Diba? Mga leishmania. Next, which of the following is the basic composition of Bacillus anthracis capsule? Okay, remember class that most of the capsule are made up of polypeptides and polysaccharides. But Bacillus anthracis capsule is made up of D-glutamic acid. Siya lang ang capsulated nga lahi iyahang composition sa capsule. So the correct answer there is D-glutamic acid. Mycolic acid is usually associated with Mycobacterium na organism. Next, 38. It is known as hay bacillus. Hay bacillus, that is your Bacillus subtilis. Okay. When you study, also try to write or summarize mga common names of the different organism. 39. Which Corynebacterium species is most common? Corynebacterium associated prostatic valve endocarditis in adults. So that is Corynebacterium jaecium. Corynebacterium diphtheriae, again, that's more likely respiratory. Ang kani silang duha, si ulcerans is more likely bacteremia and septicemia. Pertussis, C. pertussis, wala talagang C. pertussis class. So ito yung possible answer. 40. Which characteristic pattern is observed when Listeria monocytogenes is grown and incubated at room temp, not on a body temperature? Since you know that Listeria monocytogenes is a motile type of organism or bacteria, so yung characteristic niya class when grown on a semi-solid agar medium that is incubated at room temp is umbrella-like pattern. They only catalase negative, gram-positive, non-spore forming that produce H2S. So alam niyo na to, pag H2S and it's gram-positive, that's erysipelotric rochopathiae. So this one, which of the following is mismatched? So all of them class are mismatched. So recall, sino yung group A, sino yung group B, sino yung group C, at saka sino yung group E. I think most of you got correct answer for this. 43 suggests QC organisms for citrate agar slant. So you have there Klebsiella pneumoniae and E. coli are the QC organisms for citrate testing because your Klebsiella will produce positive, your E. coli will produce negative. If Shigella and E. coli, that's both negative. E. coli and E. coli, that's both negative. Morganella and Escherichia, that's both negative. So this is the best answer. Klebsiella and E. coli. 44, in a nitrate test, reduction is demonstrated by the development of red color following the addition of letter D, that's alpha-naphthalene and sulfolinic acid. Alpha-naphtol and potassium hydroxide, this is not the correct answer because these reagents are being added when you do test methyl red and Brogg-Puskauer testing, your MRBP. Say paraphenyl diaminobenzaldehyde and your amyl alcohol, this is the composition of your COVAX reagent. So this is used for identifying indole reaction. Nenhydrine and acetone are used for another testing. So ito yung possible answer, no? Magiging pag nitrate testing. Also, if ever class, no, mag-negative ang mga Enterobacteriaceae sa nitrate test, you can confirm it using zinc dust. Zinc dust. So after addition of zinc dust, if nagya po yung red color, then that's positive. If wala, then negative. Muller testing is for the detection of decarboxylase activity, which is dependent on the alkaline pH shift in the medium. So this is the correct answer. For this 2 questions review, or 3 questions rather, review your biochemical test na activity or module. ONPG testing is most useful when differentiating. Correct answer is Shigella species and some strain of E. coli, because your ONPG class will detect if it's late lactose fermenter or true lactose fermenter. At 47, at what pH does mostly methyl red test becomes positive? So the correct answer is 4.5 na pH. 48, in a test urease production, ammonia react for which of the product? So the correct answer is ammonium carbonate. Okay. Decarboxylation of amino acid, lysine, ornithine, and arginine, a result formation of, the correct answer is amide. Lysine, ornithine, and arginine are amino acids. Lysine iron agar showing purple slant and blackened bot indicates, the correct answer is Salmonella species. Sir, wait, blackened bots meaning 2 organism ang possible man, sir. Your Salmonella and your Proteus, di ba? So, Salmonella and Proteus, in order to differentiate the two, remember, your Proteus class are deaminase positive. Meaning, pag deaminase, di ba, sa atong discussion last katong face-to-face, dapat kung deaminase iya color, red, iyahang slant. Okay? So, positive man siya for deaminase, dapat red. Since the question is purple slant man, so the correct answer here is Salmonella species. 51, Klegler iron agar differs from triple sugar iron agar in the use of sucrose, because your Klegler iron agar class only contains 2 types of sugar, lactose and glucose. Wala siyang sucrose. But it has the same results with your TSI. 52, care must be taken when identifying biochemical isolate of Shigella because Serological cross-reactions occur with what organism? The answer is E. coli. Okay? Your E. coli class and Shigella may share the same biochemical results, no? And also some of the E. coli strain class or types have the same appearance with E. coli. So, dapat i-identify gud siya using motility testing. So again, Shigella is non-motil, E. coli is motil. Which genera of Enterobacteriaceae are non-motil at 36 degrees Celsius? So you have there, question, di ba kanina, anak ko, Shigella, Klebsiella, and some Yersinia species. Your Escherichia, Edwardsiella, Enterobacter, Proteus, Providentia, Morganella, Serratia are all motil. Cold enrichment in feces in phosphate buffered saline prior to subculture onto enteric media enhances the recovery of Yersinia enterocolitica. Remember class, no? Pag cold enrichment and the choices is most likely enterics. Ang answer agad ana is Yersinia. Particularly Yersinia enterocolitica. That is why your Yersinia enterocolitica is one of the possible blood contaminants sa imuhang blood bag. Kay mutubo man sila on a refrigerated temperature. Your Listeria also will require cold enrichment for growth. Okay. 55. An isolate of E. coli recovered from stool of a patient with severe diarrhea should be tested for which sugar before sending to a reference lab. So, if ever class, the E. coli, ang isolate na siya severe diarrhea, i-test na siya usually using sorbitol fermentation. Because E-hextrine will ferment sorbitol. Ang the rest nga strain wala. Shigella also will not ferment sorbitol. Okay? So, for this, the correct answer is sorbitol. That is why class, if in your readings, I think you will encounter McConkey added with sorbitol or your SMAC. So, siya. Number 56 to 65 is another case. So, Carlisle gave birth to a healthy girl. A mother came from a rural area, was good in health, and had received prenatal care. At home, the energetic infant scratched herself along the left thigh, resulting in a slight reddening, which the mother noted and treated with antibacterial ointment. Later that week, the skin of the infant and legs had started to peel. The infant was taken to a clinic where infection was examined, culture were taken, and treatment was initiated with tetracycline and nafcillin. So, the exfoliation of the large areas of the skin from different areas may suggest, So, among them, ang pinaka-close dira nga answer is the scalded skin syndrome. Okay, you have their skin as your clue. Toxic shock syndrome, usually this is bacteremic in nature. Obviously, this is not viral infection as well as fungal infection. Bacterial culture taken from wounds or abscess on an infant will most likely the presence of, if your answer is skin scalded syndrome, remember, skin scalded syndrome is only caused by Staphylococcus aureus because of the exfoliative toxin. Okay, this is number 58. The exfoliation is caused by exfoliative toxin. Number 59, strain differences of the causative agent may be distinguished by, the correct answer is coagulase test, because we want to know which strain of staph or which species staph. So coagulase test is the best answer for this. Positive result for the test being answered sa imong 59 is clot or agglutination. Effervescence, that is for catalase testing. Yellow or acid, that is for sugar or fermentation. And capsular swelling for Kilong's test. 61, proteolytic enzyme produced by suspected organism agent that causes dissolution of clot. So you have there already dissolution of clot na word. So among the 4 choices, si staphylokinase lang ang may function nga dissolution of clot. Si lipase, it will dissolve lipid components, the fats. Si coagulase will form clot. Hyaluronidase will dissolve hyaluronic acid. 62, the suspected organisms grows well in which culture medium? So you have there, kibalo man kakasti staff, so mannitol salt agar is the best answer. Okay, although staff will grow on chocolate agar, but this is not the best answer for this question. Maconchi, it will not really grow because maconchi contains some inhibitory agents for gram-positive organism. Cetrimide agar, this is a selective culture for Pseudomonas for the detection of pigments. 63, increased dosage of tetracycline may lead to these abnormalities except. So if you recall your antibiotics, no, antimicrobials na discussion, your tetracycline may cause hepatic damage, kidney, and yellowing discoloration of the teeth, example, biliary obstruction. Nafcillin antibiotic is an example of which type of cell wall inhibitors? Okay, the correct answer is penicillinase-resistant penicillin drugs. Nafcillin is not part of beta-lactam drugs because ang common example sa mga beta-lactam drugs are your cephalosporins. Glycopeptides, this is not part, nafcillin is not part of glycopeptides. Ang pinaka-common example sa glycopeptides are your vancomycin. Which of the following toxin produced by S. aureus responsible for community-acquired MRSA or methicillin-resistant Staphylococcus aureus? So the correct answer is your PVL or your Panton Valentin leukocidin toxin. 66. Which toxin released by B. cereus or Bacillus cereus that causes diarrhea that is commonly associated from fried or boiled rice? So the correct answer is emetic toxin. Duha lang ang toxin na i-release ni Bacillus cereus class, si diarrheal o si emetic toxin. And these diarrheal and emetic toxin are part of enterotoxin. Which of the following tests is specifically for anthrax? So among the four rather, Ascoli test ang pinaka-specific for anthrax. 68, which of the following forms of anthrax that shows black eschar and malignant pustule? So you have their clue already, black eschar. Eschar is only seen class as skin. So therefore, since skin man siya, cutaneous is the best answer. Okay? Pulmonary, katong Wolters disease, gastrointestinal, if it will results to gastrointestinal pain, abdominal pain, injection is different as well. 69, which colonial types of C. diphtheria is the largest? You have their clue, the largest. And daisy colonies, daisy head colonies. So kung ang largest class, si gravis ragyod na. Okay? Munang gitaw siya gravis because grabe o largest. The rest, they have their own description. So try to read the remaining descriptions of the remaining choices. But remember, largest colonial type of C. diphtheria, gravis. Pag daisy head colonies, it's still gravis. 70, in vitro toxicity test done to diagnose C. diphtheriae. The correct answer is ELIX test. Okay. Sixth test, this is for Streptococcus pneumoniae. Ascoli test, again for Bacillus anthracis. Whittle's test, this is for Salmonella serotyping. Okay. So correct answer is ELIX testing. 71, which of the following organisms are generally positive for beta-galactosidase? So this is the enzyme, no, that will test if it is true lactose fermenter. So among the four, only E. coli class ang naay beta-galactosidase ni enzyme. Okay? So the rest wala silang beta-galactosidase enzyme. 72, because your E. coli is a true lactose fermenter organism. Salmonella and Proteus are non-lactose. Shigella may be lactose fermenter, but this is a late lactose fermenter organism. 72, method of serotyping Shigella used in the clinical laboratory is based on the O antigen class, meaning the outer membrane of the organism. Diba? Review the structure of the gram-negative organism. So the O antigen is present on the outer membrane. Number 73, given the following result, which organism? So sa TSI pa lang, you will already know how to rule out, no? So acid-acid gas with negative H2S. Proteus vulgaris is K/A, so rule out. Salmonella typhi is also K/A, so wala siya labot. So ang nabilin na lang si E. coli o si Klebsiella. So E. coli is acid-acid with gas, with H2S. Klebsiella pneumoniae also with acid-acid and sometimes with gas. Now you go to the IMVIC reaction. No. Sa IMVIC reaction, indole positive, methyl red positive, Bog-Frescauer negative, and citrate negative. Most likely that is E. coli because for Klebsiella class that is negative for indole, negative for methyl red Frescauer, positive for methyl red, positive for Bog-Frescauer, and positive for citrate. So si E. coli mo'y positive, positive, negative, negative. Remember, si Klebsiella negative, negative, positive, positive. So the correct answer is E. coli. Number 74, given the following result, which of most likely the organism? Okay. Sa Maconki alone pa lang class, you already know which possible organism ang ma-rule out. It's colorless and non-lactose fermenter. So colorless and non-lactose fermenter. So meaning you will rule out no Enterobacter cloacae. Because Enterobacter cloacae, di ba? It's lactose fermenter. So sa IMVIC reaction, dili ka sure kay napakay-tulo remaining. You go to the test nga sure ka nga positive ni si vulgaris o si Proteus. So remember, di ba, your Proteus and vulgaris is H2S positive and phenylalanine deaminase positive. So since negative pa ni silang duha, Proteus vulgaris and mirabilis are already ruled out. So ang mabilin na lang class is si Serratia marcescens. Okay? So ingon ana mag-answer. Number 75, given the following result, which of the following organism is most likely the causative agent? So I think the same, no? Test sa 74, but this time you have POD positive and H2S positive. So among the choices, ang POD positive dira o H2S positive kay si fruit juice kaning duha. Si Serratia o si Klebsiella ma-out naman sila. Klebsiella ma-out siya because colorless colonies kani siya colored. Klebsiella again, remember, it's a lactose fermenter. Serratia marcescens, although it's a non-lactose fermenter, but it's not an H2S positive. So you go for vulgaris and mirabilis. So how will you identify if it's vulgaris and mirabilis? With the use of indole. Okay? Indole positive si vulgaris, indole negative naman si mirabilis. So there. Positive ba ng indole? So that is vulgaris, the correct answer. 76. Colonies of the on CIN. Remember, class CIN agar is a selective medium for Yersinia, and only Yersinia enteropolitica on a CIN agar will appear ng kaning colonies, bullseye colonies. So that's the correct answer. Yersinia enteropolitica. Number 7, which of the following organism is positive for string test? String test is usually used to test whether the organism contains capsule. Okay? So for string test positive, ang pinaka-positive nila diri na capsulated, ang pronouns kaayo ang capsule class is the Klebsiella pneumoniae. So therefore, your Klebsiella pneumoniae may also be positive on a Neufeld-Skellange test. What do you call the pigment produced by Serratia? So, pinaka-common, the correct answer is protegucin. 79, which of the following cellular structure is the H antigen? Remember your basics, no? H antigen, that is your flagella. Capsule antigen is your K antigen. O antigen, tutong sa cell wall. Not cell wall, the O, the outer membrane. 80, which among the E. coli strain is responsible for the profuse diarrhea among HIV patient? Remember class, pag HIV patient positive, that is enteroaggregative E. coli, your EAEC. Okay? Pag HIV positive patients, EAAC. Kani, for most likely traveler's diarrhea. Kani, ang EHEC, pag makakita og mga bloody diarrhea. EIC, pag EIC. It's still not common, no common diarrhea, but still with abdominal pain. 81. So as I mentioned, traveler's diarrhea, no, that's your ETEC strain. Verotoxin 1 is also produced by which Shigella toxin? The correct answer is Shigella dysentery. 83, which of the following is mismatch? So all of these are Klebsiella species. Klebsiella authene causes arthropic rhinitis. This is correct. Klebsiella rhinoscleromatis, this causes granuloma on the nose from the word rhino. No, that's correct. Klebsiella granulomatis, can cause donovanosis. Klebsiella pneumoniae will not cause atypical pneumonia. Instead, it will cause typical pneumonia. Atypical pneumonia is associated with Mycoplasma pneumoniae. So 84, again, this one, class that is Mycoplasma ang atypical pneumonia. 84, Kelong's test is used to identify which Streptococcus species. So already know na Kelong's test is for capsule. So among the 4, si Streptococcus pneumoniae ang capsulated. So that's the answer. 85, you have their beta hemolytic streptococci and the following results. So CAM test negative. So dili na pwede answer nimo si group A. So mabili na lang si group B, group B, Enterococcus faecalis, and then group A and group D. Then Bile solubility get negative and hippurate hydrolysis positive. So group B are positive for hippurate hydrolysis. So wrong na. So mabilin si C o si D na lang. So in order to differentiate the two, you can have bile isculin and 6.5 salt. So enterococcus group will have 6.5 salt and bile isculin positive, while your non-group A, non-group B, and non-group D streptococci are negative for both tests. 86. Still beta hemolytic streptococci given with this reaction. So you have their CAM test positive. So CAM test positive, only group B ang CAM test positive. The rest are CAM test negative. 87. Gram-negative diplococci recovered from an MTM plate giving positive reactions for identified as. The correct answer is all of the above. So gram-negative, all of them are gram-negative and all of them can be recovered on a modified Thayer-Martin agar, and all of them are positive for oxidase. Remember, Neisseria are positive for oxidase. 88. Gram stain of urethral discharge showing extracellular and intracellular gram-neg bacilli. So the correct answer for this is Neisseria gonorrhoeae. So M. catarrhalis, Neisseria meningitidis. M. catarrhalis is most likely for respiratory infection. Neisseria meningitidis is usually associated with meningitis, and N. lactamica is just a normal flora. So 89, again, this is a gram-negative diplococci, no, with the following reaction. So glucose positive, sucrose negative, lactose negative, maltose positive, fructose negative. So you have their glucose and maltose nga positive. So the answer is Neisseria meningitidis. Neisseria gonorrhoeae pag glucose positive lang. Neisseria zika pag glucose and sucrose lang. Neisseria flavescens class pag negative siya to all sugars. Again, it does not mean no na if positive flavescens will produce fermentation sa fructose. Pag Neisseria flavescens, negative na siya tanang sugar. So sir, negative pan po ang moraxella sa tanang sugar. How will you identify Neisseria flavescens from moraxella? You perform Dinase test. Dinase, D-N-A-S-E. Positive for Dinase test is moraxella, negative naman si Neisseria. 90. Beta hemolytic streptococci are divided into different lung field based on, you already know this, group-specific cell wall polysaccharide antigen. 91, a positive CAM test reaction is produced by group A or group B streptococci, which is your Streptococcus agalactiae. 92, a solid coagulase test is a screening test because it will be confirmed using tube coagulase testing. Okay? Because it will, your solid coagulase, this is slide coagulase supposed to be siguro. Sige, I will give this as a bonus. Your slide coagulase test is just a screening test class because the positive test will be performed or confirmed using tube test. Okay? So your slide coagulase will measure the bound coagulase. Confirmatory ang tube class because again, remember, there are some Staphylococcus species that may give positive result to slide coagulase. For example, your Staphylococcus lugdunensis. Your Staphylococcus lugdunensis can be positive for slide coagulase, but never for tube coagulase. 93, Neisseria gonorrhoeae can be differentiated, okay, by Neisseria zika. So you have there fermentation of glucose, maltose, and sucrose. Although maltose is not part, no, rarely. So if na word na sucrose, then that's the best answer for that. 94, on a blood agar plate, organism producing clear zone around the growing colonies are described to be, so you have their clear zone, na colonies. So that is beta hemolytic. Walay delta hemolytic lasa. Gamma means no hemolysis. Alpha meaning partial hemolysis. But nowadays, we do not use gamma hemolysis. In European mga journals class, you will not find the word gamma hemolysis. They will just simply write no hemolysis at all. Culture media of choice for cultivation of Neisseria. So among the 4, chocolate agar. Why chocolate agar class? Because your Neisseria organism will use the iron component found in the chocolate agar plate. 96, the enterotoxin produced by certain strain of coagulase positive Staphylococcus is destroyed by boiling? No. Identical to dermonecrotic toxin? No. It's highly antigenic. Enterotoxin are not highly antigenic. But correct answer is it causes one type of bacterial food poisoning. So 97, a beta hemolytic catalase positive, gram-positive focus, which is coagulase negative, by slide, which of the following is most appropriate action by identification of these organisms? So if ever you have slide negative test, always confirm it with tube coagulase testing. Ayaw siya i-diretso og report class as coagulase negative staph. Always perform the confirmatory testing. That is why in some laboratories class, they intend to do tube coagulase directly rather than performing slide coagulase. So 98, Landsfield classification of strep bovis. So that is group D, non-enterococcus. All Neisseria are positive for the enzyme oxidase testing. They are also positive for catalase nga enzyme. 100, a pure culture of blood agar plate grew grayish white, beta hemolytic, and give the following reaction. So you have their PYR positive, and the rest is negative. So the answer is Streptococcus pyogenes. Hindi pwede si agalacti because agalacti class is CAMP positive. Hindi naman pwede si pneumoniae kasi si pneumoniae is never a beta hemolytic strep. Si this agalacti naman is PYR negative and heparate positive. Group 101, a preschooler attending develop fever, severe sore throat. So it's a beta hemolytic streptococci. Your clue here is sore throat and it's beta hemolytic. So this is a good the possible answer for this is your Streptococcus pyogenes because siya man yung organism associated with pharyngitis. 102, middle-aged woman cut her scalp while working on her attic and there is purulent discharge, white colonies. Clue here is white colonies of clump cocci, which no hemolysis and recovered, and in bed recovered, it's susceptible to novoviacin, another clue. And also failed to clot plasma. So failed to clot plasma pa lang, S. aureus is wala na siya. So next, susceptible to novoviacin. So among the 3 remaining, only Staphylococcus epidermidis is susceptible to novoviacin. Staph prophyticus and hemolyticus are resistant to novoviacin. 103, phenylethyl agar is used in media 2. Your phenylethyl agar class is a selective medium, that will inhibit the growth of a gram-negative bacteria. Because your phenylethyl agar selective medium for gram-positive organism na siya class. This is commonly used if ever we want to isolate only gram-positive organism from a mixed culture. 104, positive result for COMS test, that is arrowhead of beta hemolysis. 105, ethyl hydrochloride susceptibility or your optokin test. So it's a presumptive identification of your Streptococcus pneumoniae. So this is the chemical name of your optokin. So for 106 to 110 is another case. So you have their 4-year-old patient who presented her pediatrician early summer with a 3-day history of vomiting and bloody diarrhea with mucus fleck diarrhea. She had several episodes of vomiting, approximately 10 movements per day. Problem were described as painful, and parents reported the patient had moderate abdominal pain. She had fever and chills. The patient had decreased fluid inputs and urine outputs. She had no recent travel or camping history, no recent change in diet, no hope is similar with illness, no history of exposure to bad food. She was cared for at home. On a physical examination, alert, vital signs were normal, stool guaiac positive was sent for culture. 3 days later, the laboratory reported non-lactose, do you have there the clue? Non-lactose, non-motile, H2S negative organism that grows on a McConkey from a patient stool specimen. So what is the possible disease of the child? So salmonellosis, traveler's diarrhea, and urdiasis. So salmonellosis, wala. Hindi siya associated with salmonellosis, no? Because again, salmonellosis, H2S negative man siya. Traveler's diarrhea, she don't have any history of travel at all. Giardiasis, remember, this is a protozoan infection. Hindi man na-mention ang any protozoan dito, so automatic cancel. So the best answer is bacillary dysentery. Since you already know that the answer is bacillary dysentery, so the possible organism is your Shigella species. And what is the possible source of infection? Shigella species or Shigella bacillary dysentery class is only acquired human-to-human transmission. So from the stool, fecal oral route. Based on the McConkey growth, what is the expected TSI? So on that McConkey, you have non-lactose fermenting and non-motile and H2S negative. So non-lactose fermenter, so you have their K over A. So meaning non-lactose fermenter, glucose fermenter. Kani glucose fermenter, lactose fermenter and glucose fermenter. Kani non-lactose fermenter, non-glucose fermenter. Kani lactose fermenter, non-glucose fermenter. So ito yung possible answer. Ideally, the specimen of salmonellosis on a 3rd week of infection is urine is the correct answer. Pag 1st week class, stool, 2nd week blood, ang 3rd week urine. Based on an IMVIC result, what test differentiates C. frondi from C. cosseri? So, the correct answer is indole testing. What is the other description of Proteus mirabilis on BAP? So you know this, diba? From the case, the odor of Proteus is burnt chocolate odor. 113, which strain of E. coli has negative results to MOG test? The answer is EHEC strain. 14, all are oxidase negative except Remember, class, only Plesiomonas species under Enterobacteriaceae are oxidase positive. The rest are oxidase negative. So, pag makakita gani og taas-taas na case about enterics and you have their oxidase negative ang result, always think of Plesiomonas species. 115, select method, choice of recovery of anaerobic from a deep abscess. So, review your specimen collection. So, pag anaerobic class bacteria, always use needle aspirate. Okay? You will not use cotton fiber because, again, cotton may contain fatty acids that may kill bacteria. Skin snip also will not be used because you're exposing the tissue sa oxygen. And swab also is never been used for anaerobic culture. So ideally, pag anaerobic culture, it should be an aspirated specimen. 116, if a clean catch midstream urine was processed, which of the following is indicative of true UTI? So, pag 100,000, mo na ang true UTI class. If ever E. coli, it should be more than 100,000 ang colony count. But if ever class, the isolate is Staphylococcus saprophyticus, di ba? Your Staphylococcus saprophyticus is the secondary causative agent of E. coli, secondary causative agent of UTI. At least 10,000 colony forming unit is considered important. So number 17, Staphylococcus saprophyticus is differentiated from Staphylococcus epidermidis with the use of, again, novobiocin antibiotics. So in order to differentiate the two, you will use novobiocin testing. Staph epidermidis is sensitive, Staph saprophyticus is resistant. Which of the following tests will rapidly differentiate micrococci from staphylococci? So the answer is modified oxidase, since both of them will form catalase positive. All of the following are culture media used to facilitate the growth of C. diphtheria, except, so all of these, Pie, Slant, Tyndale's agar, and Luther's serum are selective culture medium and special medium for C. diphtheria, except for blood agar. Your blood agar is an enriched culture medium. 120, which of the following is partially acid-fast gram-positive organism? So, pag partially acid-fast organism nga gram-pos class, think of nocardia. Okay, nocardia, yun na siya. Which of the following gram-positive bacilli show pink colonies after 24 hours? And then it's gram-positive. So for this, the correct answer is Rhodococcus. Rhodococcus equi. All of the following are protective exotoxins ni siya class released by Bacillus anthracis except tissue factor. Tissue factor is never a virulence factor. Little factor, protective antigen, and edema factor are part of Bacillus anthracis virulence factor. 123, which of the following is specific for Listeria monocytogenes and diagnosis? So again, Ascoli is for Bacillus anthracis. Will Felix, this is more of rickettsial diagnosis. Widal, this is for Salmonella. So the remaining answer there is Anton. So Anton is for Listeria monocytogenes antibiotics. I think Anton's test is an ocular test. 124, which bacteria under Enterobacteriales is typically associated with water harboring fish or turtle that it rarely causes systemic infection among patients with underlying liver disease? Pag makabasa gani og ang mga class fish and turtles and part of Enterobacteriales, always think of Edwardsiella. Okay? So, Enterobacteriales, mga fish and turtles, from fish and turtles na origin. So, the answer is Edwardsiella. Na sila. So, this one is Edwardsiella tarda. It's only one, I think, significant species under Edwardsiella. 125, all Enterobacteria are catalase positive and motile except for Shigella dysentery. The correct answer is true. Okay. That's it. Questions? Clarifications? Do you have questions? Boy. Do you have questions from the previous test? So expect class 9 ato ang mga test ninyo sa mga upcoming exams. Okay? So this is high order thinking skills since you are already third year students. Okay? So you need to analyze the problem, kung say possible answers, and you need to know how to rule in and rule out possible answers. Because ganyan naman kayo mag-take ng board exam. Sa board exam, all of them plus are possible answers. So at least kabalo mo unsaon pag rule in or rule out. Okay? And microbiology class will never be an easy subject yun na siya. Anang subject sa microbio, when you go to medicine, microbiologists, you will also have another set of subjects na microbiology sa medicine. So, dili gyud na siya ma-easy nga subject. In medicine, it's still part of board exam subject. Okay, so you need to read. All of the notes are given to you. Recordings are provided to you. So, if you have questions, please ask directly to your teachers. Okay, para ma-address diretso. Any questions? Any clarifications? Wala? Good morning, sir. Oh, na mga question. Good morning, sir. Can we ask for some configuration? Yes. Okay, so if you don't have any questions or clarifications for the 110 questions, is that yung question? Kapila gani ang week infection sa blood? Blood is usually on the first week, second week ang stool, ang third ang urine. Okay? If wala na questions, if none, then thank you for coming everyone. And I hope you learned something from these rationalizations. No? So have a great day, everyone, and see you next week. Goodbye and good morning.
Bye, sir. Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir Carl.